Hi everyone. In section 3.3, we're going to talk about what we call the cell envelope. And I don't use that term envelope a lot, but this book does. And when I'm referring to the cell envelope, we're talking about the bacterial cell wall and the cytoplasmic membrane, which is right inside. When you learned about eukaryotic cells, you learned a lot about cytoplasmic membranes. Every cell, whether you're prokaryote or eukaryote, has a cytoplasmic membrane, the phospholipid bilayer. The Not all cells have a cell wall. In fact, human cells don't have a cell wall, but bacterial cells do, plant cells do, and we're going to expand on that in this lecture. I'm going to come back to this section, and I'm going to head down here and just talk about the concept of the cell wall. And what I just want to clarify is what accounts for, or what's the importance of the bacterial cell wall. Now, we're going to talk about osmosis in a future lecture, and you have probably already learned about osmosis. It's one of those things that's really important in biology. And you might remember, you don't have to remember all the details of osmosis right now, but hopefully you remember the ideas. If you put a cell in a hypotonic solution, one that's got a higher concentration of water outside than inside the cell, then water is going to move into the cell. And this happens naturally. And in our bodies, we have a mechanism to keep the fluid outside of our cells and inside of our cells at the same concentration of salts and sugars and things. So our cells are in an isotonic solution. But bacteria can end up frequently in hypotonic solutions. So water is going to move from the outside of the cell into the cell, and you need a rigid wall. You need a strong wall to prevent that cell from popping when you put it in things like pond water or bathtub water or, lots of natural environments. So cell walls are really important structural parts of the wall. It says relatively rigid, and it has this stuff in it called peptidoglycan, and we're going to come back to that. So let's go. In your objectives, it says discuss the difference between a gram-positive and a gram-negative wall, and the book starts going on about that down here. I think it's easier to talk about that with the pictures in front of me, so I'm going to go up here to these pictures. And it says differences in cell envelope structure. There's actually two distinct groups of cell walls, not just envelopes, but the cell wall in particular. And they're called gram-positive and gram-negative. Now, when we look at bacterial cells that have gram-positive or gram-negative walls, we end up staining them in a certain way. And you can see it right here that the gram-positive cells end up staining purple when we look at the cells under the microscope, and the gram-negatives end up staining red. Now, I know that probably looks pink to you, but I always say purple and red because then I can go purple positive, and then red by default is negative. So gram-positive and gram-negative. Now, what we can do is we can stain cells to see if they're gram-positive or gram-negative, and it helps with identifying the bacteria. And that sounds like, well, gosh, if there's thousands of kinds of bacteria, what's the big deal if you just divide it into two groups? But think about if you were trying to tell somebody about a person. Oh, I met this person. Well, one of the first things somebody would want to know is, are they male or female? And when you identify that characteristic, you've just eliminated half the people. So gram-positive and gram-negative is a really important first step to identifying bacteria, and it tells us some other important things that I'll tell you about in a minute. So if I look at a bacterial cell, so this is highly magnified, and this is there's a cross section where we're just looking. Here's some cytoplasm. So here's the inside of the cell, and then you have this thin layer, the cytoplasmic membrane. And here's the gram-positive picture. Here's the gram-negative. So let's talk about gram-positive first. In this simple diagram, it's showing you here's the inside of the cell, here's the cytoplasmic membrane, and now the wall is made primarily of this thick layer of what we call peptidoglycan. And I know that sounds like a fancy word, but it's an important word in microbiology, and it's one you should commit to memory. And so here's a more detailed diagram, we're going to talk about cytoplasmic membranes in a couple minutes. This probably looks familiar, that phospholipid bilayer with proteins in it. There's that cytoplasmic membrane that all cells have. But now here we are in our bacterial cell, our gram-positive cell, that has this thick layer of peptidoglycan. And peptidoglycan is this protein and carbohydrate mixture of molecules, and it's pretty sturdy. And it's only found in bacteria. It's a unique compound. You notice that it's got some other things here, but for our purposes, those aren't critical to your understanding of gram-positive and gram-negative. So the first thing you want to remember, first two things about gram-positive. They stain purple in the gram stain, and they have thick peptidoglycan. Now, when we look at the gram-negative wall, here we have our cytoplasmic membrane. And they have it in the diagram here. All cells have that. But notice the peptidoglycan layer is a thin layer of peptidoglycan. And so in this diagram, there it is right there. There's that thin layer of peptidoglycan. Thick in gram-positive, thin in gram-negative. And then the other thing the gram-negative wall has is this thing called an outer membrane. So here you have another phospholipid bilayer. And that phospholipid bilayer is what we call the outer membrane. And that outer membrane is very similar to the normal cytoplasmic membrane, except in gram-negative cells, it's got another molecule that I want you to know embedded in it, and it's called lipopolysaccharide, or LPS. So the gram-negative wall has a thin layer of peptidoglycan, an outer membrane, and in that outer membrane, there's some LPS molecules, lipopolysaccharide. Obviously, these molecules are made of lipids and polysaccharides. And I'll tell you about their importance in just a minute. So look at those pictures, a little bit about the cell wall. It goes into more detail about peptidoglycan, and I don't go into all that, the detail about the structure of that, or how it's made and all. Here we got gram-positive and gram-negative. And again, there's more detail here about gram-positive cells than what I just told you. What I want you to know is what I just told you so far. Gram-positive stain purple in the gram stain, and they have thick layers of peptidoglycan. The gram-negative cells stain red in the gram stain, and they have a thin layer of peptidoglycan and an outer membrane that has LPS. Here it goes into a little bit about how the gram stain works. In week 5 lab, I believe, we actually learn about the gram stain, and we do virtual gram stain. So this is good background for that. And what you see is when somebody does a gram stain, this is what it looks like under a microscope. There's 4 different stains you use. So you put your bacterial smear on a slide, and then you're going to dry it onto the slide, and you're going to expose that slide with the bacteria on it to some dyes. And the first stain you put on there is called crystal violet. And crystal violet just sticks to the cell walls. And so right here it's showing you the crystal violet goes into that thick layer of peptidoglycan in the gram positives, and it goes into that thin layer of peptidoglycan in the gram negatives. So if you looked at it under the microscope at that point, both gram positives and negatives would look purple. The second ingredient to the gram stain is what we call Gram's iodine. And so we put that on there. We rinse off the crystal violet. We put in Gram's iodine. And what the iodine does is it sticks to the crystal violet. And here we have, in this thick wall, we have the iodine going in and sticking to those crystal violet molecules. And you create these great big molecules. And it's kind of funny because the crystal violet was small enough to get in. The iodine's small enough to get into that wall. But when they clump together in that thick gram-positive wall, they're too big to get out. Okay, so it's kind of like they got in the doorway. They're small enough to get in the doorway, but when they clump together, they got too big to get out of that doorway. So they're stuck inside the wall. Now, because the gram-negative wall, peptidoglycan is so thin, it doesn't have the ability to make these big clumps of crystal violet and iodine, so they don't get trapped in that wall. And the third step is something we would call a destain, where you use alcohol to rinse it out. So here we rinse the gram positives, and those big clumps of crystal violet and iodine stay in the wall. So the wall remains purple. But those big clumps weren't able to form in this thin wall, so the gram-negative walls lose their color when you destain it with alcohol. And so that's what they're trying to show you here, that after the alcohol treatment, the gram-negative cells are colorless and the gram-positives are still purple. Finally, we add a fourth stain called saffronin. And when you add that to these bright purple gram-positive walls, it doesn't show up. You don't see the color. But when you add it to this colorless gram-negative, they turn red, or what it looks like kind of here, pink. And that's just a way to make the gram-negative cells show up so you can see them under the microscope rather than looking at colorless stain. So by the end of the gram stain, your gram-positive walls are purple, your gram-negative walls are red, and that is determined by the thickness of the peptidoglycan. So, so far we've learned that the cell walls are important in preventing bacterial cells from popping, but they're also a useful tool they're a useful target for us to help identify bacteria using gram stains. Let's see. I think I had something else to tell you. Yeah, give me a second. I'm trying to see where it is in here. Right here, there's something else about the cell wall when it's talking about peptidoglycan down here. And it says the feature of the cell wall provides the cell wall is a target for antibacterial drugs. And I'm talking about penicillin in particular. And I just wanted to point this out because when you think about making drugs, you have to find something that kills the bacteria, but doesn't kill the host. So when you take an antibiotic, you want a drug that kills bacteria, but doesn't kill you. And cell walls are a really great target because what you want to do is you want to think about what do the bacteria have that the host doesn't. So bacteria have peptidoglycan, and it turns out penicillin it targets peptidoglycan. We don't have peptidoglycan, so penicillin won't target our cells and it won't kill them. So another benefit to understanding bacterial cell walls is that we can use them for drug targets. And it turns out that gram-positive cells are more sensitive to peptidoglycan more sensitive to penicillin because they have this thick layer of peptidoglycan. Gram-negative cells tend to be naturally more resistant to penicillin. And that's because they have the peptidoglycan, but they've got this outer membrane that protects them from the penicillin from getting in. So if you have an E. coli infection, which is a gram-negative bacterium, you probably wouldn't use penicillin to treat it. But something that's gram-positive, like streptococcus, might be more likely to be treated with penicillin because it's more sensitive. So now if I go back to my gram-positives and my gram-negatives, I'm going to put a lot of P's together. I'm going to say gram-positive stains purple in the gram stain, and it has a thick peptidoglycan, and it's penicillin sensitive. It's naturally sensitive. We're going to talk about bacteria that become resistant to drugs, but this one usually is naturally penicillin sensitive. Penicillin can kill it. So our gram-negatives have a thin peptidoglycan, stain red with the gram stain, have an outer membrane with LPS, and are more naturally resistant to penicillin. Okay, so if you get your gram-positive and your gram-negative straight there, you'll be in good shape. The other thing, I want to say one more thing about gram-negative cells, and it's down here. It talks about that outer membrane with that lipopolysaccharide, that LPS. And what I want you to know is that LPS is actually toxic to us, and it's something that we call an endotoxin. Now, I'm not going to ask you a lot about that right now, and it goes into a lot more detail than I will. But later on, when we talk about how bacteria cause diseases, endotoxins are important. So kind of tuck that into the back of your mind to remind yourself that when you're infected with a gram-negative bacteria, this LPS is going to have a big role in making you sick. So we'll talk about that later on. One of the things I always like to remind you about bacteria is every time I tell you something, there's probably an exception. There's so much variety in bacteria. So here I have one exception. It says non-typical cell walls. Well, it turns out some bacteria don't have cell walls, and they're totally different. They have other ways to protect themselves from osmotic pressure. But one I wanted to talk about was one that has what we call an acid-fast wall. And here we do what we call an acid-fast stain. The kind of bacteria that for which we use an acid-fast stain are in the genus Mycobacterium. And Mycobacterium, they cause diseases like tuberculosis and leprosy. These are really bad diseases. I know all the nurses have to get TB tests to see if you've been exposed to it. It's really contagious. It's a respiratory disease. Well, mostly respiratory disease, but it's in the genus Mycobacterium. And it turns out that Mycobacteria don't have a gram-positive or gram-negative wall. They have this wall that we call acid-fast, and it's kind of waxy. And what it does is it makes it really resistant to drying out. So bacteria in the genus Mycobacterium, like the ones that cause tuberculosis, can remain on surfaces sometimes for 2 weeks and still be able to cause infections. And when we go to identify that, we do something called an acid-fast stain. And I think we do that in one of our virtual labs. The last thing I wanted to mention in this section was just to remind you about cytoplasmic membranes. So remember we said the bacterial envelope is the cell wall, which is on the outside, and then there's a cytoplasmic membrane on the inside, just like every other kind of cell. The main function of a cytoplasmic membrane, whether it's in bacteria or a eukaryotic cell, is it controls what goes in and out of the cell. It regulates what goes in and out of the cell. It's selectively permeable, meaning it lets some things go in, some things pass through without letting others pass through. And it looks a whole lot like a eukaryotic membrane. It's a little bit of differences, but remember it has phospholipids and proteins. And so if we go back and look at here, this all should look familiar of that. There's a cytoplasmic membrane, that phospholipid bilayer with proteins. That controls what goes in and out of the cell. The cell wall is more of a basket. It's got bigger spaces and molecules can go in and out. It's the cytoplasmic membrane that controls what's in and what's out of the cell. And that's where I'm going to stop on section 3.3.